Yes, sir.
Okay, I think this is the second last class. So next
Yes,
week we have, sorry, it's the second last class, right? When you have your examination, when you have your exam for this NPTEL, the final certification exam,
ma'am, on 28th this month.
28th. Okay, fine. So we'll be starting with the topics covered in the last week, that is week 11. So, week 11 was about the applications of biotechnology in social welfare. So, basically, in this week, we have dealt with the applications of biotechnology in plant and animal sciences. So, up till now, in this course, we have studied how to create a genetically modified organism. Okay, we have studied about cloning, we have studied about the various vectors, various expression systems that we can use, bacterial system, eukaryotic system, yeast, insect system, all this we have studied. And then we have studied how to express our gene of interest. For that, we need an expression vector. Then we have studied that after the expression of our gene, which like a protein, then how to purify it and how to extract it, how to purify it. So, for that, we have dealt with the downstream processing, the downstream applications, which involves the extraction and the purification of our product, of our recombinant protein. So, we have studied about various techniques such as electrophoresis. We have studied about different kinds of chromatography that can be used. And then we have studied that, okay, we have cloned our gene, we have expressed it, we have now even purified it. What is the next thing? Next thing is to characterize it. And what involves in the characterization of our product? Characterization involves finding the molecular weight, finding the functional groups that are attached to it. So that all we can do with the help of electrophoresis also, like with the help of gel exclusion chromatography, we can find out the molecular weight. And then with the help of various spectroscopic techniques such as UV visible spectroscopy and infrared spectroscopy, mostly we dealt with these two, we can characterize our product. Now that our product is there, okay, so up till now we have studied right from the creating of a recombinant gene and then right from creating the recombinant gene up till purification and up till processing it. Now we'll be seeing, now that we have seen in the last week, that what are the applications of those products that have been already made in biotechnology. So in week 11 lecture 1, we have studied about the role of biotechnology in plant sciences. Okay, so in plant sciences, we have studied about the biotechnology the recombinant technology and biotechnology is used for insect control, sterile male insects, insect resistant plants, in herbicide control, in making herbicide resistant plants, then in disease resistant plants, then in plant tissue culture. What is plant tissue culture? How to prepare the culture media? What is explant, how to prepare the explant dishes, and all these things. So very quickly we'll just go through, we'll have a quick recap of what we have studied in our lecture 1, and then we'll move to the questions. So like this is not like a very difficult, this was not a very difficult week in terms of the concepts, because you just have to go through about the various applications of biotechnology. But more important, I think it was in the lecture 3 where you have studied about monoclonal antibodies. So monoclonal antibody is an important part, and some points like basics of some of the very famous examples of in BT cotton, and some examples that were given here. They are important in terms of your competitive point of view, competitive exams point of view. So you can just, it's like a very easy topic. Plant and animal biotechnology is a very easy topic, and it is also very scoring in terms of your exams, like in terms of your exams for your gate or for your CSIR. So in those terms, it is easy also for you to learn, and it is very scoring also. So now we'll first go through the what we have studied in our first lecture. So this is from where we started. So we started by creating a recombinant clone. We started by having our gene of interest, that we isolated from the whole genome, and then the plasmid, the vector in which we want to put it, and then we did the digestion, we ligated it, we transformed it, we screened it, and then we got the clone, and then we expressed our clone, and then rest is what all we have studied. So biotechnology has applications in various fields such as in agriculture, drug delivery, pisciculture, poultry, vaccines, and so on. Then if we talk about the applications of biotechnology in plant sciences, so in plant sciences, so what are the problems? So first of all, for creating any drug, for creating any medicine, first thing we need to have a problem. Like when you're doing any experiment, when you're doing research, first thing is to have a problem. So when you have a problem, then you think of a solution, and for that solution you make a hypothesis, and then you make a hypothesis, and then you plan your experiments. So for having any kind of drug, any kind of product, or any kind of thing, first you have to look what is the problem. So what problems does plants face? So plants face a problem from insects, that these insects, they kind of eat the plants and deteriorate the quality, and therefore controlling the insect population during agriculture or plant culture is important. So for the control of insects, what can be the strategies that can be used? So basically they thought that the best strategy would be to make the male sterile, so that when this sterile male undergoes mating, there is no production of offspring. Okay, so and because it's easier for to produce a sterile male than uh, trying to go for a sterile female. So then for the production of sterile male, so what they did, so they basically took the approach of some kinds of radiations and some kind of chemical treatments that made this male sterile. Now when these sterile male insects are spread over the areas, then the offsprings production was decreased. Obviously the offspring production will be decreased because these males are sterile. So the examples of this is ball wheel. Just a minute. Okay, so this is one thing of having a sterile male. And then what you can do, one thing is that you make insects in such a way that they are not able to produce much population, and therefore you are controlling the insect population. Okay, but there is no biotechnology much which is involved here. But the problem here was that you are making your insects sterile. So these insects are not just spreading on the area where you want. Obviously, they will spread to other areas as well. And somewhere where these insects might be required, it will go to those areas also. So it will have more negative effect than creating some positive impact. So another way then scientists thought that why not just not think about the insect population? Why not we make our plants only so much resistant that insects cannot harm them? Okay, so a very classic and a very popular example of this is BT, Bacillus thuringiensis, BT cotton. I think BT cotton, I think everybody, or if you haven't studied up till now, you will study. It's a very important and a classic example. So basically, here comes your genetic engineering. Here comes your whatever we have studied, the application of it. That if you are making your crop genetically altered in such a way that it is resistant against the insects, okay, so what kind of genetic engineering was done here? So basically what they have done is that they have genetically modified the plant so that it expresses a toxin which will kill the insects, but it is safe for human consumption. That means what we have studied that you can take any gene of interest and that you can express, okay, with the help of genetic engineering. So the gene of interest that they took was a toxin. So this toxin is harmful for the insects, harmful for insects, but it is not harmful to the humans. So by this way, by expression of this toxin, basically the population, whenever there is an insect, the population of insect will be reduced, because now this particular crop is genetically modified. So that particular harmful toxin is released from Bacillus thuringiensis. So Bacillus thuringiensis BT is a bacteria which secretes an insecticidal toxin. Okay, so this was a strategy. So basically, they have used genetic engineering because they have expressed the BT toxin in their somatic cells. Okay, so when insect feed on the plant, toxin reach to the stomach and cause internal bleeding to kill the insect. That means one way is that you spray the BT toxin, or with the help of genetic engineering, you need not to spray it. So, can you tell me what would be the disadvantage of spraying a particular insecticidal resistant in comparison to genetically modifying it within the plant? What would be the advantage of having a plant which is genetically modified in comparison to spraying of a toxin? Same toxin, can anybody tell me?
Ma'am, the
Will there
plant
be
which was sprayed with a toxin are not much more sustained for the long period of time as compared to the plant which were genetically engineered for resisting to the for resistance to the insect. Means it can for long period of time it can protect itself from insects as compared to non-engineered plant.
okay? So what do you mean by long period of time? As in, suppose if like here in BT toxin, so this BT toxin is sprayed, is expressed in the somatic cells. So what do you think that the expression of somatic cells will have effect on its longevity? Like it is in the somatic cells. So can you just elaborate it? Like what do you want to say? Are you talking about the half life of the particular enzyme when you are spraying it, and so because of many other environmental factors, it kind of starts degrading, or but when it is inside the plant itself, then it is always there? Are you trying to focus on that?
Yes, ma'am. Ma'am, I mean that it is genetic so that it can produce a certain toxin throughout its lifespan, and it can be passed down to next generation that specific characteristics to resist itself from those insects. But I think the next generation would not be resistant of the normal plant to protect itself from insects.
Okay. Okay, and also that when you are spraying something, you cannot control spray. Like it will go to the areas where it is desired, and it will also go to the areas where it is not desired. So, therefore, it is always good to have the modification within the plant, but then it also has its own cons, because it is it's like that we are playing with the nature. So it might have some consequences in the longer run in the food chain, in the food web, and it might have some other harmful effects that are not visible in 1 to 2 generation, probably in the longer run. So that is still a debate. That is why creating recombinant products is always under ethical concern, especially in India, and even more when it is when you are taking some kind of protein or some kind of gene from an animal. So it comes under a lot of ethical restrictions, also because there are people who are vegetarians, so they are reluctant to take a recombinant product which has a gene from, let's say, pork, let's say from pig or from any other animal or any animal per se. So there are very uh, a lot of constraints, a lot of restrictions when it comes to creating genetically modified organisms, and probably that is the reason why in India you don't have so much recombinant products freely available in the market. Anyways, then we come to herbicide control. So in herbicide resistance, so basically here it is the problem. So like the one I was saying, that suppose you have herbicides. Okay, so you spray herbicides on a particular area to kill the weeds. Okay, so basically weeds are the unwanted plants that grow along with your crop. Okay, so you want only your crop to grow, not the weeds to grow. Why? Because these weeds are also competing with the nutrients, and these weeds are not required. So suppose if I am growing a plant here, this is my plant that I am growing, this is my plant which is important for me, and there are so many weeds which are growing all over the places. Okay, now whatever nutrients, whatever minerals, whatever water I'm putting it for the growth of this plant, it is getting divided between this and the weeds also. Now these weeds are just good for nothing, so I want to kill these weeds. So what I thought of doing, that I will just spray some chemical herbicides over it. Now what will happen, these herbicides are what these herbicides are not just selective for these weeds. Okay, they are selective to a class of plants, or they can be non-selective and they can kill all the plants. So what will happen, that if I spray herbicide over this area, so herbicides are the chemicals that kills the weeds, that kills the herbs. So this will destroy the weeds, but this will also destroy, or this will also damage or have a harmful effect on my plants of interest. So that means herbicides, these herbicides, they are not only killing the weeds, they are killing the good plants also. Because you cannot control their action, right? Because they have a non-selective action, or even if they have a selective action, they have like a range. They're not completely specific to one kind of plant. So that is why this strategy is not a very good strategy, because by this way you are damaging your own plants, and also you are damaging the soil, because you are putting so much chemicals that you are damaging the soil as well. That area of soil which is right now grown by weeds, when you are spraying so much of chemical herbicides, the soil is also absorbing those herbicides, those chemicals. And when you think after some time, if you think that you will grow some other plants over this area, so those plants will take up those chemicals, because earlier this region was sprayed with chemicals. Therefore this spraying of herbicide is a very bad idea. So scientists thought that let's try to put some genetic engineering in it and try to have a different or better variant. So what they did was that there is glyphosate. So basically, glyphosate is the first herbicide which was designed to kill weeds. So what it does, it that it interferes with the biosynthetic pathway of the, sorry, huh. So it interferes with the biosynthetic pathway of the aromatic amino acids like phenylalanine, tyrosine, tryptophan. So it interferes with the aromatic amino acid biosynthesis pathway. Okay, so basically the plants which are treated with glyphosate, so these plants cannot produce these aromatic amino acids, and therefore these plants die. So what scientists did was that they genetically modified the crop plant so that they are able to get these aromatic amino acids. Okay, so that is what they have written, that the genetically modified crop plant is designed with an alternate pathway to supply the aromatic amino acid in the compensation of the EPSP, basically in compensation with the inhibition which is happening because of the glyphosate. And second strategy was to use an alternate form of EPSP that is resistant to glyphosate inhibition. So basically they are making the plant in such a way that even when you are spraying your glyphosate, the plant is not getting killed, the plant will survive. Because the problem that the agriculturists were facing, that when we are spreading the weedicides, sorry, when we are spreading the herbicides or weedicides, our plants are also getting killed. So they have thought that we should make our plants in such a way that our plants will not be killed when we spread the herbicides on them. So for that they have modified their plant in such a way that, okay, when you are spreading glyphosate, the problem with glyphosate was that glyphosate inhibits the aromatic amino acids pathway. So why not to make a plant in such a way that it can have some other alternative sources of aromatic amino acid synthesis? Or what we can do is that our plant is resistant to EPSP only, our plant is resistant to glyphosate only, and so there is no point of having an alternate amino acid pathway. So that is what they have done. So, but again, it this is a technique that can protect our plants, but I would not say that it is a 100% sure shot technique, because it is not completely eradicating our problem. The next strategy is to make a disease free plants. So for creating disease free plants, what they have did, so basically you can see here, this is the list of disease resistant plant. So this is basically what all they have done. So it all of these are the examples where they have used the genetic engineering approach by expressing a certain gene, a certain product, which will make it resistant to some kinds of disease, which will make resistant to some kind of uh, other kind of abiotic stresses or biotic stresses. So the basic idea, the basic concept is expression of a recombinant gene, expression of a outside gene into your in your plant, so that it gains a selective advantage. That selective advantage can be making it disease resistant, that selective advantage can be making it resistant against weeds or insects or whatever. You're just creating a selective advantage over others. Okay, so basically these are all the different techniques that they have used. I will not go in such detail because we have to cover a lot of things and we have to do the questions as well. So then we come to the other important aspect of plant science, biotechnology in plant science, is your plant tissue culture. Okay, so plant tissue culture, basically, you just when you're just going through the steps, so you have a plant. Explant is any part of the plant that is used for the growth. It can be leaves, it can be stems. So it is explant means any part of the plant that will ultimately help in the callus formation, and that callus formation will help in the generation of roots and shoots. So I think someone has asked a question. I think he left the chat or something. I don't know. But Narayan Rao has asked a question in the beginning, that which part of plant is able to generate whole part in its tissue culture. So basically the part of the plant is, it can be any plant, it can be any part. Okay, mostly people use leaves. So that part of the plant that you are using for tissue culture, that is known as explant. For example, you use leaves, you can use other parts also. So I'm not a plant person. So if anybody wants to add up in something but which I am saying, you can, because I am from animal background, but this is the basics, so I can explain you. Then you have your plant. So then you have your explant. That means any part of the plant that you want to use for your tissue culture. Let's say I am taking leaves. Okay. Then what I have to do? I have to sterilize it. Why I have to sterilize it? Because plant is in it's an open, suppose, or even if it's not in open, so you have to sterilize it so that you are getting rid of all the microorganisms which are over the plant, and that may hamper in the growth, that will help for a better sterilized environment, because that can cause contamination. So, whenever you are using tissue culture, whether it's plant tissue culture or an animal tissue culture, it is very important to have a sterile environment, to have a sterile explant, because that will lead to contamination. And if you have contamination, even if you are getting positive results, you cannot say that it is completely because of your growth or because of the growth of some other contaminants. Let's say some microorganisms. So, contamination can give you false positive results many times. So that is why it is very important to have a sterilized environment in tissue culture, especially in animal tissue culture. I would say that in animal tissue culture is even more prone to contamination and infections. Anyways, so then you have your explant. Okay, now you will inoculate your explant in the media. Okay, so why you have to inoculate in the media? What will media provide? So media will provide us the basic nutrients, salts, basic glucose, sugar sources, all those things that are needed for the growth of the plant. All those things are in the media. So then when you provide the media, then there will be formation of the callus. After callus formation, you will have roots and shoots, and then you can transfer it. You can just transfer the plant into the field. So then you have a lot of instruments that can be used. So cork borer is basically used for creating discs. So basically, so when you have leaf and you want to take the explant, you basically create discs, small round discs, which is basically used for your callus formation. The explant, if this is your leaf, with the help of the borer, you will just take a small disc and then they will use it. Then, so the basic procedure of plant tissue culture, it involves the preparation of the culture media, sterilization, then surface sterilization of the explant, then explant disc preparation that I told you, small disc supposed from the leaf, then its inoculation, then incubation, and then acclimatization and hardening. So this is your preparation of tissue culture media. You have, mostly they use MS media, and these are the various sources, which various constituents of the MS media. These are all important for the proper growth and development of the plant. And then you had very, I think here also, I think the next there was a very beautiful video also. So this is all the basic steps you just have to go through, nothing much to explain. You have to sterilize it and properly wash it. You can see these are the discs here, and then you inoculate. Here is the callus formation taking place. You just have to go through all these things. Now we'll just quickly try to solve some questions, and then we'll move to what is implant. Chalo, abhi pehle first we'll do some questions. So the first question is, irradiation of boll wheel was achieved with the help of removing the female population from the area, killing the offsprings as soon as they were born, irradiation of the egg to make them infertile, exposing male insects through radiation to make them infertile. So what do you think is the answer? Can anybody tell me the answer to this question? Option D. Okay. So option D is exposing male insects through radiation to make them infertile. Yes. So we have already just discussed. Even if you don't know, see, you have to look for the strategy to solve a question. So even you have absolutely no idea about what this bowl wheel is about. You just look at the, if you have the basic concept, if you have just a basic knowledge of what we have studied, removing the female population from an area. Do you think it is practically feasible to remove female population or to remove a male population, especially if they are, let's say, insects? It is not very easy and possible. Again, killing the offsprings as soon as they were born. Is there any biotechnology involved over here? No. Irradiation of the egg to make them infertile. We have already discussed that making egg infertile is little bit difficult because the females go through a typical cycle, and there is which is a little bit more complex, exposing male insects through radiation to make them infertile. So if you even have just a little bit idea that disease insect resistant involves making male insects infertile through some kind of chemical treatments or radiation, you can solve the question. Now next question is how bt toxin helped in creating insect resistant plants. Bt toxin was sprayed onto plants so that insects were killed. Bt toxin was genetically modified that caused killing of the plants. Plants expressing Bt toxin caused death of the insects. Or option D, Bt toxin could not provide resistance against bacteria. So what do you think is the answer?
Option C.
Option C, that is plants expressing Bt toxin cause death of the insects. Yes. So why? Because what can you explain a little bit of the way or the mechanism through which it happened? We have studied that it created some kind of toxin in the intestinal area, in the stomach area, and therefore the plant died. Okay, we come to the next question. What is the mechanism of action of glyphosate? It catalyzes the conversion of shikimate 3 phosphate to EPSP. It helps in the production of aromatic amino acids. The EPSP is modified and cloned so that it can surpass the glyphosate mechanism of action, or none of the above.
Ma'am, option A.
Option A, you are saying that it catalyzes the conversion of shikimate 3 phosphate to EPSP. Okay, any other? Just a minute. Okay, so another we are getting as none of the above. Is this the answer of this question 3? For the question 3, are you saying none of the above? Karthik? Okay. Yeah. So we have two options, A and D. So the right answer is D, and we'll see why. Okay. So basically, you said this option, which is it catalyzes the conversion. No, this is the pathway this pathway basically this pathway helps in the production of aromatic amino acids. So glyphosate is inhibiting this pathway. Here it is written that it interferes with the biosynthesis of aromatic amino acids. Okay, so the for the biosynthesis of aromatic amino acid, this pathway is involved. So basically, glyphosate it inhibits the enzyme EPSP synthase. Okay, and this enzyme catalyzes the conversion of shikimate 3 phosphate to EPSP, and therefore it leads to the production of aromatic amino acids. So, glyphosate is inhibiting this pathway. Glyphosate is inhibiting the conversion of shikimate 3 phosphate to 5-enol pyruvate shikimate phosphate EPSP by inhibiting its enzyme, that is EPSP synthase. So glyphosate is a herbicide. Okay. So to kill the herbs, to kill the weeds, it targets the production of aromatic amino acids that occurs in the plants. So for targeting the production of aromatic amino acids, it targets on this pathway, and to target this pathway specifically, it targets the enzyme which is EPSP synthase. Okay. So we had the options That it catalyzes the conversion? No, it interferes with the conversion. So it cannot be the answer. It helps in the production of amino acids? No. Then the APSP is modified and cloned so that it can surpass the glyphosate mechanism of action? No, it is not surpassing. So the options D is correct. The next question is, the carbon source in plant tissue media, specifically we're talking about the MS media, is glucose, sucrose, glycogen, or all of the above. What is the carbon source in MS media?
Ma'am, all of the above.
No. We're talking about in MS media, mostly the carbon source.
Glucose.
It is
Um,
sucrose.
yes, it is sucrose. So you can just see here, mostly the carbon source in MS media is sucrose. So the correct answer is B. So our next question is, surface sterilization of plants can be achieved by sterilization using 2 in 20, washing using distilled water, washing using 70% ethanol, or all of the above. What is the answer?
D. All of the above.
Yes. The correct answer is D. All these strategies are used. Okay. Now we'll come to the lecture 2. In lecture 2, we have studied about the applications of biotechnology in animal sciences, where we have studied about production of therapeutically important proteins, production of human insulin, gene therapy, and some vaccines. Okay, so very quickly we'll just revise what we have studied and then we'll go to the questions. So again, in animal technology, in animal sciences, when we talk about, so just thinking about the areas where biotechnology can be used. So biotechnology has applications in artificial insemination and in embryo transfer. So basically, what do you think that this artificial insemination and embryo transfer will lead us to achieve? Like, ultimately, what is the goal? What we want through this artificial insemination, through this embryo transfer? What is our ultimate goal? Can anybody tell me? What is the ultimate goal? Why we are doing artificial insemination? Why we do embryo transfer in animals?
Ma'am, because of reducing the birth defect or chances of carry on the pregnancy or not having miscarriage.
Yeah. So ultimately, what these will lead us? These will give us better
Improved
yield,
variety.
better yield. Then, as someone said, improved quality, improved better variety of whatever we want, and also kind of disease free lines also. So basically, um, artificial insemination and embryo transfer. So if you just look up into it, so this artificial insemination is now in our country, very uh, profitable, not profitable, but it is a very widely used technique now. It's a very widely used technique, especially in the parts where agriculture practices are more. So I am from a college, from an institute where they mostly focus on the animal biotechnology aspects. So I personally have seen farmers coming and purchasing the semen straw. So semen straw is basically used for artificial insemination. Okay, semen straw, like a normal regular straw which is filled with semen um, of a good of a good yield. That, I mean, that will get a good yield of a good breed. So So this artificially inseminating your cattle, your cows with the help of semen straw is now in some areas of India is becoming a practice, and farmers they do buy these straws and they try to artificially inseminate, okay, the males. So I'll just go through quickly the steps for the artificial insemination. So basically, the superior breed male. So you have breeds which are superior, superior, and as in the sense that their semens has sperms which has a good motility rate. Okay, so good motility rate means better fertility chances, and also uh, they have the genes which yield to a better yielding breed. Okay, so that is a superior breed which has a good better motility rate semen sperms, and which ultimately lead to the production of breeds which are of a good yield. So, yeah, like it is written that the quality of the sperm, such as motility and the number, is checked through a microscopic observation. So, you just have to do, so nothing you have to do, you just have to take a straw, just have to put a drop of semen into it, and you just have to observe under the microscope. And then you have to, by visually, you can see how motile are the sperms, or what is the sperm number, sperm count, and that all contributes in creating in a superior breed quality. And then so you basically when you are taking semen for creating for artificial insemination for creating your semen straw, it is not just the raw semen, it is mixed with some other things such as milk, yolk, glycerin, etc. And then very important is that it's very actually very important. I don't know whether any of you have had the chances of seeing this artificial insemination in person, but it is very important to know that when you are doing something artificially, you have to synchronize everything. Okay, something which is happening in nature and something which is happening in artificially, in vitro or in artificially, here artificially. So in nature you need not to synchronize anything. So when cattle mate naturally, so they are already in heat, so their estrous cycle, so like in humans we have menstrual cycle, so in cattle they do not have menstrual cycle, so they have estrous cycle. Okay, so I will not go into much detail of the estrous cycle, but estrous cycle, just like in menstrual cycle, you must have studied that there is a point in the almost at the mid of the cycle where the point where ovulation is going to take place. Okay, and when mating happens during that time, then only the chances of pregnancy are high. Not at every time of the menstrual cycle a woman can conceive. So same goes for cattle as well. So when you're trying for artificial insemination, you cannot just take a straw and you cannot just artificially inject it into a cow or into a female without having any prior knowledge of the time of or the exact time of its ovulation. Because if the ovulation is not happening place, if the egg is only not releasing, then what is the point of injecting semen into it, because there will be no fertilization. So that means that you have to have the knowledge of the estrous cycle of the female before doing artificial insemination. Okay, so you have to know the exact timing of ovulation, and at that time only the semen is injected into the female reproductive tract, and therefore you need skilled people for it, for performing the artificial insemination. So artificial insemination is one application of biotechnology in animal sciences, but it requires the prior knowledge of the estrous cycle of the females. That is one point you should always understand and always remember. Then we come to okay, so these are some of the advantages and disadvantages, that is okay, that you can go through. Then we come to embryo transfer. So there is one thing. first you should understand the term which is superovulation. So superovulation means that generally what happens, so in each estrus cycle, in every each cycle, so each estrus cycle leads to the release of one egg. Okay, each estrus cycle leads to the release of one egg. But when you are doing artificial insemination, you have purchased a straw which has cost you some bucks. Now you are using though that straw for artificially inseminating your female, and you just have one egg. So the chances of reproduction, the chances of fertilization is very low. So now what is done now, that so the female is given some hormonal treatment which leads to the production of or the ovulation of more than one egg in a single cycle. That means the female is not just ovulating one egg, it is super ovulating more than one egg. So because of super ovulation, you have several eggs in just one cycle. Okay. So there are many different ways by which you can take the advantage of several eggs. Suppose if you have a very good superior breed female, and you are able to fetch more than one egg or super like more than one eggs in one cycle, you can harvest those eggs and then you can transfer it to different other females. So that is what embryo transfer is about. So basically what you do is that using an here it is written that using an AI procedure, superior breed male is used to fertilize the egg. Once the embryo is formed, it is transferred into another female. So basically what is happening, so what is the logic? The logic is that normally what you do, what you can achieve, you can only achieve one fertilization in one estrus cycle. But here what you are doing, that you are using artificial insemination also. That means you have a check that the semen that I am injecting into the female, it is of good quality, and you have taken a good quality, superior quality female also. And because of super ovulation, it is producing more than one eggs. So ultimately in one cycle you are able to get more fertilizations, you are able to get more fertilized eggs, and then you will collect all those fertilized eggs using a catheter, and then you will transfer all those fertilized eggs in different females. Now the transferring of those eggs in different females, those females need not to be of very superior quality, because what the DNA is, you have already taken the DNA from that egg, the DNA has come. So need not to be a superior quality female for during the transfer. Okay, so this is how this takes place. But again here it is very important that the donor animal and the recipient, it is very important. See, these are very small intricacies when you read about any technique. You embryo transfer, okay, you have to take a egg from superior female, transfer it into a maybe not such superior inferior female, that's it. But the important point here is that both donor and recipient animals, they must have a synchronized estrous cycle for successful embryo transfer. That means that here I am talking about the estrous cycle of the superior female which has produced the fertilized egg, and the estrous cycle of that uh, low breed female which is going to keep the fertilized egg, their estrous cycle should also be synchronized. Okay, so it is not here in the course, but if anybody who is interested, so there is a complete procedure of synchronization of estrous cycle of the donor and the recipient. So that is beyond the scope of this lecture as of now, because of the time constraints we have to cover other points also. But there is a complete protocol of synchronization of estrous cycle. Okay, there are many papers also, in fact, if you want to study that how you can synchronize the estrous cycle of a donor and a recipient female. Anyways, now we'll come again. So here you can see superovulation, then artificial insemination, then recovery of the embryos, then either you can transfer the embryos to another female, or you can store the embryos. And it is very important that before transferring you have to just check the embryos. So basically there is a classification of the embryos. The classification of the embryos is mostly based on the type of follicles that are present in the embryos. So there are many kinds of follicles of different grades, which are known as grade A, grade B, grade C, grade D. So grade A and B follicles, if there are more in the ovary, then it will lead to a better fertilization. So anyway, that is let's not get into that much detail. Then we have the applications of biotechnology in medical sciences. So up till now we have studied that biotechnology has application in plant sciences. It helps in creating disease resistant, weedicide, herbicide resistant, insect resistant plants. Then we have studied that biotechnology has applications in animal sciences. In animal sciences it is used for artificial insemination and embryo transfer, and there are many more applications as well. Now we come to medical sciences. So in medical sciences, it is used for the production of some therapeutically important proteins, gene therapy, vaccines, and monoclonal antibodies. Okay, just a minute. Okay, fine. So, for the production of therapeutically important proteins, like for example, insulin. So this is all just what we have already studied. That what you have to do, you have to take your gene of interest. Suppose here we're talking about insulin. So insulin is a dimer of two chains, A and B chains. So individually you clone your A chain, individually you clone your B chain, you transform it, then you express it, then you have the peptides, then you recombine it, then you get the recombinant insulin. So like this way, using the basic genetic engineering approach, sorry, yeah, so using the basic genetic engineering approach, there is a list of the therapeutic proteins which are in use in the recombinant technology. And again I'm telling you that this table is very important. Okay, this is an important table. So when I say important, I'm not just referring to a certificate, this thing, certificate in exam. My focus is to tell you the important topics that are important for your competitive exams as well. Because if you are interested in higher research, higher studies, if you want to go for masters or PhD, then you have to give your GATE or DBT or CSAR, and there these some of the points that I have been discussing in each and every class. They all are like the hot topics. So here this table is important for your gate. And this table is mostly important for your gate. So there might come one or two questions in DBT as well, but for gate it is important. So in gate you have your plant biotechnology and animal biotechnology topics also, if I remember. In gate or in DBT, I think in both of them or in either one of them, you have plant and animal biotechnology topics. So whatever it is there in this lecture 11, all the points are important for your gate and DBT point of view. Then gene therapy. So gene therapy means that suppose here these are the therapeutic proteins. Okay, so these are the therapeutic proteins that you intake, that you take. And there is another approach that involves that you need not to take any therapeutic proteins. There will be changes in your DNA only, so that there is no need of taking proteins from outside. So they are correcting the very base level of the disease. So suppose if there is any mutation that has happened in the DNA, okay, if there is any kind of mutation that has happened in the DNA, so if gene therapy involves correcting that mutation so that your DNA will not have that mutation. So ultimately, earlier your DNA was producing a defective protein. Now because of this gene therapy, your DNA is being corrected, and now the protein that it is producing is completely functional and it is good. So that is your gene therapy. So basically, see, what I am telling is like a very high end therapy, which is not very, I don't know whether it is done in India or not, but yeah, in foreign, especially in US. Now it is a very new technology that has come up to that. So I think you all have heard about sequencing. So now people are trying to have personalized vaccines, personalized medicines. So personalized medicine depends upon uh, like the sequence of your DNA. That means they will sequence your DNA, and according to your body, according to your DNA sequence, according to your body needs, they will prepare medicines that are specific for you. For example, if I take a paracetamol or if I take a dolo, so that is a general medicine. Okay. So that is a general medicine that everybody takes. But some medicines work very well for some people. Some medicines they don't work that well for some other people. Why? Because everybody's immune system is different. Everybody's response to vaccines, to medicines, to therapeutic agents is different, because everybody is has a unique DNA and a different immune system. So some medicine which can be very good, it can be a very good medicine and it can work wonders on my body, cannot have any effect on somebody else, or can have some toxic effects of on somebody's else body. So personalized medicine is a very new concept that has come, which means that according to your DNA, according to your DNA sequence, they will give you medicines. Okay, so in gene therapy, when we coming back to the gene therapy topic, so basically gene therapy can be done in two ways. Okay, so first one is somatic gene therapy, and the other one is your germline gene therapy. Okay, so somatic gene therapy means that whatever genes, therapeutic genes are there, they are transferred in the somatic cells. Therefore, they will not go to another generation. Whereas when we talk about germline gene therapy, so germline gene therapy means that there the required gene is transferred into the germ cells. Germ cells means your sperm or egg cells. So when they are transferred into the germ cells, then what will happen? Then the treatment, it will go to the next generation as well. Okay, so but your germline gene therapy is not a very promising approach because of the various risks and various constraints. So people mostly mostly somatic gene therapy is better choice, or more researched and a studied topic. Then these are some of the problems which are associated with gene therapy. You can just go through it. They are short lived, they can have some immune reactions, and it can disturb the host physiology and all. Then we come to vaccines. So basically, what is vaccine? So if there are two terms, can anybody tell me what is um, passive immunization, and another one is active immunization, and other one is passive immunization? Can anybody tell me what does it mean? What is passive and active? What is passive
Ma'am, in
immunization?
passive, we are giving ready-made antibodies, and in active, we are just giving the immunogen, and our body is producing the antibody.
Okay. So vaccine will come under passive or active?
Ma'am, active, artificial.
Okay. Can you repeat what you said about active and passive? What difference?
Ma'am, in active, our body is producing the antibody, and in passive, we are providing the body with ready-made antibody. And and the vaccine is an example of artificial active immunity, where we are providing the immunogen externally, and antibodies produced by our body itself.
So see, in active, you're in passive immunization, in passive immunity, so our body is producing the antibodies, right? So because of whatever exposure to the antigen, the body is producing immunity by producing antibodies. Whereas in active immunization, you're giving the antibodies itself. So the body is not producing antibodies by its own, but it is already getting the antibodies from outside. So vaccines are basically, like it is written in your slide, that they are dead or attenuated form of the organisms or maybe the proteins derived from them. In short, vaccines are basically antigens that you are giving. So when you're giving antigen to the body, will it come into active immunity, active immunization, or passive immunization? You are not giving antibodies to your body, you're giving antigen to the body.
Ma'am, it's
Are
a
you getting?
passive immunization.
Yeah. So you have to understand that basically, so let's take the example of the most talked about antigen in recent times, COVID. So uh, people got vaccinated. Okay. Some people had a good immunity against the COVID. Some people still got COVID even after vaccination, but it might not have created a very drastic response, the COVID after the vaccination. But in other people's, even after vaccination, they got a very drastic fever, and like there were a lot of symptom, many symptoms for COVID even after vaccination. So why it happened? Because different bodies have different immunity. So different bodies, they responded to that vaccine differently. In some bodies there was a lot of production of antibodies, so it gave them good, better immunity. So even after getting infected with COVID after vaccination, they didn't had much symptoms. But in others body, because of the less production of antibodies, they could not get immunity from that vaccine. So therefore they had even more like they had worse symptoms for that particular disease. So basically, how a vaccine will react in different bodies, it depends on the person's immunity. Some people will have a greater immunological response to the vaccines, other might not have that much uh, in response to the vaccines. And that is the reason why you give booster doses. So basically, when you give booster doses, so these booster doses ensure that I don't know whether you have studied or not. So there is a curve. Okay, let's not go into that detail. Just remember that once you are infected with a particular antigen, there is production of antibodies. Okay, and these antibodies have the memory of that antigen. So when again you are exposed to that same antigen, the antibodies will produce an enhanced response, a greater response against those antigens. So when you give booster doses, because your body has already experienced those antigens before, so when you're giving those antigens again, your body will produce even greater response to those antigens. So because your antibodies have memory. So like we have memory when we read something, when we see movie, when we go and when we listen some songs. So there's a memory which is stored in our brain. So when again it comes, then we can easily recall that okay, we have already seen, we have already studied, we already saw this movie, we already went to the place. So likewise, our antibodies also, they have memory. They also remember the antigen that has encountered in the body. So when that antigen encounters again, the antibodies will produce even more better response, because now they know what they have to do, they are experienced. Okay, so basically vaccines are nothing but you are giving that antigen only that is causing the harmful effect, but you have modified the antigen in such a way that it will just produce immunity which is responsible, which is required for the production of antibodies. It will not create such an exaggerated response that you can fall sick. You may have like a little bit feverish, you can have like a little bit fever for a day or two, but it will not be very harmful or as harmful as the antigen itself. So for the production of vaccines, there are different strategies that has been used. Okay, so suppose if this is I'm talking about, let's say the antigen is a bacteria. So this is a bacteria which is responsible for producing a disease X. Okay. So you want to make a vaccine against this bacteria. So what I can do is that either I can just kill this bacteria, and then I can inject it into the persons to create immunity. Or what I can do, I will not kill this whole bacteria, I will just attenuate it. Okay, so what I will do, I am just attenuating it and then I am giving it to the body. This I can do. Or what I can do is that I know that why to give this whole bacteria, when I know that inside this bacteria only this particular portion is responsible for producing toxicity, then I will just give this portion only, I will not give the whole bacteria. Okay, so I am telling you that there are different kinds of vaccines. There is killed vaccines, there are live vaccines, there are attenuated vaccines, there are subunit vaccines, there are toxoid vaccines. So these vaccines, like what I am telling you, these vaccines depends upon the different strategies by which you can give the antigen to the host body. But the logic remains the same, the concept remains the same, the ultimate goal remains the same, that the body should produce an antibody, an immunological response against that vaccine that will help the body to protect from further exposure of that antigen. Okay, this is like a very basic of vaccine. So now here comes the different types of vaccines that I was talking about. Just a minute. Okay, okay, fine. Yeah. So different types of vaccines that I was talking about. So vaccines can be of different types, like killed vaccines, where the whole organism is killed. Okay, attenuated vaccine is that you have just attenuated the whole organism, you have not killed it. Okay, toxoid means that the toxic component of that particular organism is inactivated and then it is given as the vaccine. Subunit means that you have isolated a particular protein from that toxic compound which is responsible for the disease and then you are giving it. Okay, here you are giving the whole inactivated toxic compound. Here you know that in that toxic compound, this is the protein X which is responsible for the disease, and you're giving that particular portion only. Conjugate means that your bacterial coat, it is stacked with a immunogenic protein to create a much more enhanced immune response. So basically, there are different ways and strategies in which you can create vaccines. Vaccines is also an important topic. Okay, this is also important. Now we'll just go through the questions. So the first question is, superior breed male and superior breed female are known as dam and sire respectively, sire and dams, legs and dams, dams and legs. So what is the answer?
Option B.
Option A, dam and sire respectively. Yes, option A is the right answer. The next question is, identify
Option
the correct
B.
option. Superior breed male. Yeah, sorry. Option B is the correct answer. So superior breed male is known as sire and superior breed female is known as the dam. Option B is the correct answer. Right, good. Then seventh question is, identify the correct statement relating to the embryo transfer method. Eggs are fertilized and transferred to superior females. Eggs are fertilized in low breed females and then transferred to high breed females. Fertilization occur in vitro and the fertilized eggs is subsequently transferred to superior female. Eggs are transferred in the fertilized in the superior female and then transferred to the low breed females. So what is the correct answer?
Option D.
Option D.
D.
Okay, so what is option D? Eggs are fertilized in superior female and then transferred to low breed females. Yes. So basically, fertilization, it always occurs in the superior female, which is known as dam, and then it is transferred to the low breed females. Fertilization does not occur in vitro. They are not transferred to superior females, and eggs are not fertilized in low breed females. So D is the correct answer. Then recombinant product for blood clotting is collagen 1, factor 8, pro 542, serum albumin.
Factor 8.
Factor 8 is the correct answer. Okay. Then we come to this matching. So try to see and then try to solve it. So attenuated vaccine, what should be the option? Attenuated vaccine.
Organism cannot cause disease.
Okay, so there is one answer A. Then subunit vaccine. Subunit
Ma'am,
vaccine.
sorry, for attenuated, it should be chemical or UV treatment to the pathogenic organism.
Okay, so should I'm not saying it is right or not, but I have two options for A. So you only said you only told this option, or some other student has told this option?
Ma'am, me only.
Okay, so we have just one here, A. Okay, then just a minute. Hmm. Okay, then subunit vaccine.
Organism cannot cause disease.
Okay, killed vaccines.
Ma'am,
Inactivated
I think
toxic compound.
Mm-hmm.
Ma'am, I think killed vaccine should be chemical or UV treatment to pathogenic organisms. So the organism is killed and then attenuated for organism cannot cause disease and toxide for inactivated toxic compound.
I'm getting confused. Just so you okay. So can you just give me in message, like everybody has different these things. Okay. Okay, again we'll go one by one. So for attenuated vaccines, what is the answer? Like maximum number of people, what they are saying. For attenuated vaccines, what are you saying? Tell me.
Organism cannot cause disease.
Okay. And for subunit vaccines?
Minimum allergy.
Hmm. Okay. Killed vaccine?
Chemical or UV treatment to the pathogenic organism.
Okay. And toxoid is inactivated toxic compounds. Okay. So does anybody else has any other options? If anybody else have any other this thing options, you can tell. Someone was also telling at that time. If anybody else has other this thing. So this is the correct. We'll just quickly check it. So attenuated vaccines. So basically, see, yeah. So attenuated vaccines, basically the organism's ability to cause disease is destroyed, and therefore you need to have booster doses when you are giving attenuated vaccines. Okay. So here attenuated vaccines, A, organism cannot cause disease, yes. Subunit disease, minimum allergy, yes. So basically, is it written over here also? Yes. So subunit vaccines will give us minimum allergic responses. Then killed vaccines, C, chemical or UV treatment, yes. These treatments only used to kill the pathogen. Then toxoid is inactivated toxic compounds. So this is the correct answer. Okay. So now we'll go through the last portion where there is monoclonal antibody Production and biotechnology in pisciculture. So first we'll see the monoclonal antibody. So what is the, can you differentiate between polyclonal antibody and a monoclonal antibody? What is the difference between a polyclonal and a monoclonal antibody?
Ma'am, monoclonal antibodies produced from the same B cell clone, and so obviously they are against the same antigen. Polyclonal, polyclonal is against the same antigen but from different B cells.
Mm, okay. Anybody else who wants to give a try? Anybody else? Okay. So, just a minute. Okay. There are two terms that you should remember. First is epitope and another one is paratope. Okay. So, epitope is that portion. So basically, when an antigen and antibody interact with each other, it is basically the epitope which is present on the antigen and the paratope which is present in the on the antibody that interact with each other. So when we say what is a monoclonal and a polyclonal antibody, the definition should be a monoclonal antibody is an antibody which is specific against a particular epitope, a single epitope. Whereas a polyclonal antibody is an antibody which is not specific to a particular epitope. It is specific. It can bind to a set of epitopes. Matlab it is not specific. Whereas if one antibody it can bind to three different antigens, then how you can say that this antibody is specific? No, monoclonal antibody is specific to one particular antigen. So that is your monoclonal antibody. That means mono. That means it is specific to just one antigen. Whereas polyclonal antibody is not specific to one antigen. It is specific to many antigens. So when I am talking about specific to many antigens of one particular antigen, I am specifically talking about that monoclonal antibody is specific to just one epitope, one type of epitope, whereas polyclonal antibody is specific to a number of epitopes. Okay, so when we talk about monoclonal antibody production, so basically monoclonal antibody production means that now there are two properties that are harvested in producing monoclonal antibodies. First is that you want the antibodies. So the general in general antibodies, so what was the disadvantage? First thing is that okay, they were not specific to a particular antigen. Then you have B cells. Okay, you take B cells and they you can have antibody that is specific for a particular antigen. But what will happen that it will die after some time. Uh, it will not grow indefinitely. And then you have myeloma cells, which are not specific to the antigen, but they have the property of indefinite growth. So when you are fusing myeloma cells, which has a property of indefinite growth, and B cells, which will provide a specificity to particular antigen, you will have antibody which is specific to a particular antigen also, and which has the property of indefinite growth also. That is why this technology is known as hybridoma technology, because it is a hybrid of both myeloma cells property and B cells property. So those are your fused cells. So basically what you do, that suppose I want to create monoclonal antibody against an antigen X. Let's say I want to create monoclonal antibody against an antigen X. So first I will give that antigen to the mouse. That is known as immunization. After giving that antigen, I will harvest the cells. from the spleen. I will harvest the cells, and those B cells I will fuse with the myeloma cells, and then I will select those cells that are fused cells. Because when I am fusing these B cells with the myeloma cells, I will have some cells which are just myeloma cells, some cells which are just fused B cells, and some cells which are fused cells. So I don't want just B cells, I don't want just myeloma cells, I want fused cells. So for the selection of fused cells, the medium which is used is hat medium. Again, I am telling you, monoclonal antibodies again is an important topic for your exams. So this hat medium is hypoxanthine, aminopectin, and thymidine medium. So basically, this medium is used. Why? So you have two kinds of nucleotide synthesis pathway in our body. The first one is a de novo synthesis pathway, and the another one is a salvage pathway. Okay, so de novo synthesis pathway. What is the meaning of de novo? De novo means from the scratch. Okay, so in de novo synthesis pathway, the synthesis of nucleotides occurs from the scratch. From the scratch means that from initially they will synthesize a nucleoside, then they will add phosphate, and then they will make a nucleotide, and then individual nucleotides are made, and then they will have the nucleotide synthesis. So de novo means that from the scratch nucleotides are synthesized, and salvage means that it is not from the scratch. It occurs during salvage nucleotide synthesis mostly occurs during just to maintain the proper homeostasis of the body, then salvage nucleotide synthesis pathway occurs. So this hat medium has hypoxanthine, aminopectin, and thymidine. Okay, so what will happen that I don't know whether it is huh. So what happens is this aminopectin, it blocks the de novo synthesis pathway. Okay, so de novo synthesis pathway is blocked. So when the de novo nucleotide synthesis pathway is blocked, then nucleotides will not be synthesized, and then therefore those cells will die. So B cells will anyways die after some time, because these are not indefinite cells. Okay, see, you have to understand it like this, and it is very important, huh? So if you have like a basic idea, a basic clear idea about how this selection of hybridoma occurs, then it is very easy for you to understand this monoclonal antibody technology. So this A stands for aminopectin. So aminopectin, it blocks the de novo, sorry for the writing, nucleotide synthesis pathway. But I just told you that there is another pathway also, which is known as salvage pathway. Okay, there is salvage pathway also. So what? If the de novo pathway is blocked, the cells can take the salvage pathway. So B cells can take the salvage pathway, but B cells do not have the property of indefinite growth. So anyways, they will die. Okay, B cells, they have salvage pathway. B cells, they have salvage pathway, but because they do not have the property of indefinite growth, so they will die after some time. Whereas myeloma cells, I don't know why it is not writing. Sorry. So myeloma cells, they do not have the salvage pathway in them. Okay. Salvage pathway is not present in myeloma cells. So now what will happen? That B cells are going to die because they will not grow for indefinite period. Myeloma cells will not die because they do not have salvage pathway. But the fused cells which has B cells also and myeloma cells also, they will survive, because myeloma cells will provide us indefinite growth and B cells will provide us the salvage pathway. So like this aminopectin is helping in the specific selection of fused hybridoma cells, and it will stop your myeloma cells from synthesizing. And these hypoxanthine and thymidine, these are basically used in the salvage pathway. Okay, so therefore this hat medium is used for the selection of hybridoma cells, and how aminopectin works is very important topic. Believe me, Even up till my PhD, even in my PhD coursework, I studied the same. Even in your bachelor's level, you are studying this hypoxanthine aminopectin thymidine. And even in my master's PhD coursework, I'll tell you that not many people were clear that how this happens. Now what I have told you is just like a basic gist, okay? So you can even refer to some good books, and there you can even have like a detailed knowledge of how hypoxanthine and thymidine are able to synthesize the other nucleotides, and how actually how this salvage sorry de novo pathway is blocked. That also you should know. I'm not telling here because it is again beyond the scope and capacity of this class, but you should know that how the de novo pathway is blocked by aminopterin, and how hypoxanthine and thymidine helps in the salvage pathway. These two things also you should know, okay? So this is like the gist, that hybridoma technology means that you are creating monoclonal antibodies where you have specificity also. Specificity is coming from B cells, and indefinite growth is coming from myeloma cells. And with the help of hat medium, you are able to select only fused cells, because aminopterin will block the de novo pathway, and myeloma cells do not have salvage pathway, so they cannot use the alternative pathway. B cells, they can use the alternative pathway, but anyways they are not going to survive for many generations. So in this way you will select the fused cells. Then
Ma'am, if we fuse two diploid cells, then won't we get a 4N cell, like a tetraploid cell?
so uh, can you tell me about the status of myeloma cells and B cells, the like the nuclear status?
Ma'am, both are diploid.
Mm, okay. So B cell has one nucleus, and myeloma cells.
Also have one nucleus.
Your question is very good. I have to go through it to think, but I will definitely answer it in the next class. But I think, as far as I could understand, that here the creation, even if it is even if it is getting deployed, I don't know. At right now, I don't know whether it is actually getting deployed, or they are through some mechanism they are inactivating one nucleus or removing one nucleus. I have to go through it, but even if it is deployed, I don't think that it will have like much problem here, because our interest is getting that monoclonal antibody. The diploid or haploid or like four nucleus, this will not affect the efficiency of that monoclonal antibody, okay? But to specifically see whether what is happening in the nuclear status, that I have to check. But it's a very good question. um if I have time, I can check after the class also, if we have time. Otherwise, I will answer this question. I will try to see to find out the answer of this question. And I think you should also go through, and you should also see, and all the students in the class, because he has raised a very good question, that what is happening to the nucleus status when these cells are getting fused. So just give me 5 minutes. I will try to see
Okay, ma'am.
what happens to the nucleus status. Hmm. Um, I think it will take a little bit of time for me because I have to go through papers. Mm, mm. Meanwhile, you can also check if you can get. See, You have B cells. I haven't read anything. I don't know because I have to still go through the papers. But just think about a basic logic. You have myeloma cells and you have few B cells. Okay. You have hybridoma cells. What you have? Hybridoma cells. Bees, but what you want? Do you want a complete cell, or you want specific antibodies that are produced in those cells? That we have to see. We are just, what we are extracting is the antibody, just the antibody. In a cell, there are so many things. In a cell, uh, uh, so there are so many other compartments. There is nucleus. There is mitochondria also. There are antibody production also that is happening. But are we focused on the whole cell, or we are extracting the antibodies from those cells and we're just giving those antibodies? Because it's not just about the B cells here, myeloma cells are also there. So B cells will produce antibodies. Do you understand? So B cells. So if you have read about the mechanism of development of B cells, when B cells encounter antigen, so they go under a process of clonal expansion, and that clonal expansion will lead to the production of antibodies. And these are B cells. See, it's easy now. No need to go to paper also, I believe. Okay, we can go through paper for seeing the nucleus status of whether how the nucleus status is behaving. But I really don't think so that that will have any effect here, because ultimately we are not injecting the whole cell. We are just extracting the antibodies that are produced from those hybrid cells. And also one thing that I have read, I don't know whether it is applied over here or not, but when you have some kind of hybrid which is happening, any kind of hybrid which is happening, so it also depends upon the stage at which you are taking out the nucleus. So if the stage at which you are taking out the nucleus, for example, if you're talking about a somatic or a germline cell, it is at the very beginning. Have you read about this male pronucleus thing? Have you read about this male pronucleus? Okay,
Mom, in
anyways,
context of sperm.
in the context, in general context, I'm trying to explain that when fertilization happens, at the time of fertilization also, there is a time at which the when the fertilization is happening, so one nucleus, the male nucleus is a little bit bigger, male or female nucleus is a bit little bit bigger, and one nucleus is small, and then they kind of combine with each other. So I'm not sure what is happening here, but some kind of recombination is definitely taking place. But in respective of what kind of recombination occurs over here, what we ultimately need are the antibodies. So if these are your B cells and they are exposed to antigens, then they will propagate and they will lead to the production of antibodies. Now these antibodies can be of two types. One kind of antibodies are the ones which are memory B cells, which are not secreted. These are adhere to the membrane. The other one are the plasma B cells, the plasma B cells antibodies. So these antibodies are the ones which actually uh, helps in the creating an immunological response. So when you are fusing myeloma cells and B cells, so these cells will lead to the production of some kinds of antibodies, and those antibodies are what we are interested into. This is completely my notion, my thought. As of now, I'm not saying that this is what only happening, but this is what I can think as of now is happening in the case of monoclonal antibody production. But in our next class, I will try to see if you have my email, just please remind me before the class to just look into it, because I will definitely forget, because I have so many other things also. So I might forget to read the chapter, to read about this. But if you can please remind me about this, so I'll definitely read, because it is actually very interesting to see what happens to the nuclear status when the two myeloma cells and B cells are combined. Okay, but the basic concept, the basic logic remains the same, that we are interested in the antibodies and not the whole cells. And the antibodies will be produced, and the antibodies will have the characteristics of the myeloma cells and the B cells both. And so this is what is happening. So basically we want B cells, we want such kind of B cells that are not dying, so that there is indefinite production of antibodies that is happening. Okay, the antibodies are getting produced from B cells only, but because of the fusion of the myeloma cells, these B cells are not dying now. So therefore there is more production of antibodies that is happening when in comparison to a B cell which is not fused. That is the whole concept. So just like we'll go through the protocol. Uh,
Ma'am, actually in animals they say that we cannot have polyploid cells, right? In plants we can have hexaploid, tetraploid.
yeah,
So how
in
will it
animals we
survive
cannot
if
have.
it gets
So that's what I'm
uh,
saying, that there must be a mechanism that I have to go through, that is kind of causing the killing. Killing would not be a good technical word, but somehow there is some kind of damage or destruction which is taking place, or might be there is some kind of fusion that is which is taking place, or we need to have the diploid or haploid status of the both the cells to carefully look into what happens, because polyploidy is not possible, that is not going to happen. So but how uh, it is happening, the mechanism that we have to look, that we are sure that it is not happening in animals, that happens in plants only. In animals it doesn't happen, but how it is happening, that we have to look into. So could you find anything? Could you find anything, or I think
Ma'am,
we'll
I found
discuss.
some answers. They are saying that as myeloma cell is a cancer cell, and cancer cells are used to surviving polyploidy. So that's why maybe the cells are like tetraploid or have are not diploid, but still they survive because they have those properties of the cancer cells. Hence, they can survive the state of being polyploid.
mm, Not very convinced with the answer, but okay, we can take that as one point. But that is not actually the exact answer. Just a minute. Hmm. We have to go through it a little bit more deep to understand that what is actually happening with the nucleus status, because this heterokaryon that is forming. Yeah, I think so, because what I could see here from one of the articles, so they are saying that the heterokaryon, they retains nuclei, and there is a single cell which has two or more nucleus. Okay, so single cell has two or more nucleus, and yeah, so there are two or more nucleus in the single cell, and then okay, let's not just waste time, because we have to go through the questions also. We'll discuss about it in the next class. I mean, that is now for sure. We know that there are two or more nucleus, and it has no relation with the production or the efficiency of production of antibodies, because ultimately we're not injecting a diploid diploid or like polynuclear bacteria polynuclear cell into the organism. We are just taking out the antibodies from that cell, and that only we are injecting. So we'll see that, but first we'll quickly, because we don't really have much time now, and I have still some part to cover. Okay, so what you have to do is, we have studied, it's simple now, that first you have to immunize your animal. Okay, and first you have to prepare your antigen. From here you will start, that you have to prepare your antigen. Then after the preparation of the antigen, you will give the antigen to the animal. And then you will after giving the antigen, you will isolate the B cells, or you can have the testing of the antibody, collection of the serum. This is all like basic. You just have to understand that first you have to prepare the antigen, then you have to give the antigen to the animal, then you have to isolate the B cells from the spleen, and then you have to screen those myeloma cells with the B cells, and then you will harvest those cells. Okay, and then yeah, and then the screening process is important, because the efficient screening process will only give the good recovery of your hybridoma cell lines. Otherwise you might have B cells or myeloma cells also, like the contamination of B cells or myeloma cells can also be there. And I think there was a video also in the lecture. If you have seen the class completely, there was a video also from CCMB Lucknow. I haven't seen the video yet, but you can go through the video and you can see that how the antibody generation is happening. It is not, it is just the antibody generation. You can just go through that video if you haven't gone through it. Okay, so I think these steps we have already just read. Now I think we should go to the questions. And then in pisciculture, you have again the breeding for creating some ornamental fishes or edible fishes. And then in ornamental fishes, it depends on, like we have glowfish, which has fluorescence in it. So you can have the improvement in the quality, the color, the size. And then this is all, I think this is all, you can just go through. It's not much explanatory. I think these all are just some small points that you can go through. Now we'll just solve some questions. Okay, I think, yeah. Basic question: a person with hereditary disease can be cured with the help of gene therapy, dialysis, chemotherapy. What is the answer?
Gene therapy.
Gene therapy.
Yeah. Then quickly, next question. In monoclonal antibody technology, tumor cells that can replicate endlessly are fused with mammalian cells that can produce antibody. The result of this fusion is basic and easy question. What is the answer?
Myeloma.
Hy-
Myeloma.
No. We're talking about
Hybridoma.
the result of the fusion. The fusion will create what?
Hybridoma. Hybridoma.
Hybridoma.
Hybridomes. Yeah. Then the next question is, in human, B and T cells are matured in maturation. We're talking about B cells are matured in
Bone marrow.
Thymus and bone marrow respectively.
uh, B cells are matured
Bone
in
marrow.
bone marrow, and
Bone
T
marrow.
cells are
Bone
matured in
marrow.
thymus. Okay,
Thymus.
so T for
BB.
thymus and T for T cells. Okay, next question is three options. Okay, vaccines protect us from getting infection by destroying the infective agent. Oh, I'm so sorry. Yeah. Destroying the infective agent, protecting the insect vector. Okay, sorry. Protecting
Increasing
the
the immunity
insect
of host.
vector in the environment, killing the insect vectors responsible for the spread of infection, or increasing the immunity of the host. What is the answer?
Increasing the immunity of host. D.
Yeah. Increasing the immunity of the host. And I'm sorry for the typos. Yeah. Next question is, the vaccine used to prevent tuberculosis is, it is not, we have taught, but general questions. Which vaccine is used to prevent tuberculosis? Any idea?
BCG.
Yeah.
BCG.
BCG.
Then question 15, main objective of production of herbicide resistant crops is to eliminate weeds from field without the use of manual layout, eliminate weeds from the field without the use of herbicides, encourage eco-friendly herbicides, reduce herbicide accumulation in food particles for healthy safety. Health safety. What is the answer?
Option A.
Herbicide resistant crops we are producing, so that what is
We
the
encourage
basic
eco-friendly herbicide.
sorry, can you please
Answer
repeat? You're not
C.
audible, that's what I'm saying. What is the main objective of production of herbicide resistant crops?
Option A.
Okay, and why would you say that?
Ma'am, if the plant is also sensitive to that herbicide, then we would have to carefully lay out the herbicide in such a way that it is not coming in contact with the plant. So we would need to manually lay it out. So, but if the plant is resistant to the herbicide, then we can spray it all over the field without
Okay,
manually laying it out.
so I think A is the right answer. And can you think that D would also be a right answer or not?
Ma'am, D.
D. D.
D.
Reduce
Because
herbicides accumulation.
basically what we are doing is, so what you said is okay, that is also correct, but you see, so the problem that was coming was that when you were spreading your uh, herbicides, so because herbicides were not only killing the weed, they were also going into the plants that are useful for us, that we might be using for say for human consumption. So those herbicides were also getting accumulated in the food, in the plant food that we eat. So when a plant is herbicide resistant, so that means it is not able to take that herbicide, so therefore that plant will have reduced accumulation of that herbicide. So I think D would also be a right answer. Now, like if these
Ma'am,
kinds of questions,
herbicide accumulation would mean that it's physically accumulating on it. So, I mean, if the plant is dying because of the herbicide, then of course we won't consume it. But if it is not dying and then we are spraying the herbicide all over it, then of course it will, bioaccumulate.
see, if the plant is herbicide resistant, so there are two things that we have to understand, that if this is your plant, here one thing that you have to understand is that when you are spraying something on the plant, okay, so the soil might accumulate it, but the plant will not accumulate it because it is resistant. Just because it is sprayed doesn't mean that it will take up and it will metabolize. So the herbicide will be able to show its toxic effect when it is metabolized in the plant in such a way that it creates some toxicity or toxic products. If it is just on the plant surface because it has been sprayed all over, it can be washed off. But if it is taken up by the plant in such a way that it is getting metabolized in the plant, and then its toxic product is expressing in the plant, then it is coming in the food in the consumption. So that you have to understand. Could you understand what I'm trying to say? So just spraying of something doesn't mean that it will it will be taken up by the plant. It will be taken up by the plant when the plant is able to metabolize that product, or when that product is able to cause some deteriorating effect in the plant. And that is what the whole objective of creating herbicide resistant genetically modified crops, that even if you are spraying it, even if by some means it is getting sprayed on the plant is resistant in itself. So it is, sorry, um is there any problem? Okay, so The whole objective of creating a herbicide resistant plant is that even if it gets sprayed on it, because it is resistant in itself, so it is not able to create that metabolic toxin that is harmful for the plants. Okay, so both can be correct. And uh, mm, so when these kinds of questions come in examinations, it's actually very tricky to select which option you should go through. But the basic strategy is to think of an answer which is more direct and specific. More direct and more specific, that is the key. Direct and specific. Here, both are specific, but if I have to answer, maybe I will go for the D1 because it is more direct. But that doesn't mean that A is not the right answer. A is also equally correct. Okay.
Ma'am, if a plant is herbicide resistant, that doesn't necessarily mean that it is not taking up the herbicide, right? I mean, we can provide it with the alternative pathway to solve the issue that's the herbicide is causing, but that doesn't necessarily mean that it is not taking up the herbicide, right?
It depends upon the kind of mechanism that we have taken to make it resistant to the herbicide. Okay, so that you are saying is different. You're getting confused now. See, I'll just explain you. In the first lecture only it was there. Mm, herbicide control. So here there were two strategies. The genetically modified crop plant is designed with an alternate pathway to supply the aromatic amino acid to compensate for the inhibition. You have to understand this. See, can you just repeat the question so that I can explain you in a better way?
Ma'am, I was saying that if a plant is herbicide resistant, that doesn't necessarily mean that it is not taking up that herbicide, right? Like in this example only, it is saying that it is taking up the glyphosate, but we are providing it with an alternative pathway to overcome the problem that has been
But
put up
that
by
is what the
glyphosate.
see. But that is what the negative effect of that herbicide, na? This herbicide, the negative effect of this herbicide is that that it is causing the inhibition of the amino acid synthesis pathway. That is the only negative effect. Suppose if I have sprayed glyphosate in a plant, and I have engineered the plant so that it has an alternative pathway to supply for the aromatic acid. So now the only negative effect that this particular glyphosate is causing on the plants is that it is causing the death of the plant because the plants are not able to synthesize the aromatic amino acids. It is not just about taking. See, that is what I'm saying. How the metabolism is working. The glyphosate is responsible for the inhibition of amino acids, and those inhibition of aromatic amino acids lead to the death of the plant. But here, the even though it is causing the inhibition, but because this inhibition is getting overcome by some other plant, so this glyphosate is not able to harm the plant because the plant has made some different strategy to overcome the deteriorating effect of glyphosate. So doesn't matter if the even if this glyphosate is, that is what I'm saying, that there can be two strategies to deal with the herbicide. The first strategy is that the herbicide is getting inside the plant but it is not able to create a toxic product, or the second strategy is that it might create the toxic effect a product, but this toxic product will not have any effect on the plant because plant has evolved some other strategy to overcome this toxic effect. So the strategy can be different, but the consequence is the same, that the spraying of the herbicide is not causing any effect on the plant. And that is why we are making it herbicide resistant. No, if even after making herbicide resistant, the plant is getting harmed by this the herbicide, then what is the point at the first place of creating a herbicide resistant plant? Strategy can be anything. Either you are making it in such a way that it is resistant to the glyphosate inhibition. You see, second point, few bacterial strains use an alternative form of ESP that is resistant to glyphosate inhibition. So either you are making it glyphosate inhibition resistant to glyphosate inhibition, or if you're not able to make it resistant to glyphosate inhibition, you are making some other strategy that is overcoming the glyphosate inhibition. Could you understand?
Ma'am, could you show the question again once?
See, the question is, yeah, main objective of production of herbicide resistant is reduce herbicide accumulation in food particles for healthy safety. See, even this herbicide is getting accumulated in the food particle. We are concerned with the herbicide accumulation. Why we are concerned with the herbicide accumulation? Because when this herbicide will accumulate, it will cause the deterioration of the plant. But because the deterioration
It
of
can
the,
also affect us also, right? I mean, of course, it is not affecting the plant, but if we consume the plant and this,
see, it
all
is
these
now that is like a very generalized term to say that it can affect us or whether it cannot affect us. Then it depends on the kind of herbicide that we are using, and it depends on its mechanism of action, that what it is causing. Okay, just saying that because I don't know, you can, it can affect or it cannot affect, you cannot say, because you don't know that the pathway that they have made, have they made it in such a way that it is causing the complete the production, the inhibition. If the inhibition is much lower than the production, if the production is much higher than the inhibition, then there would not be much effect. The effect would be very negligible, it would be very small. And suppose, let's say if you talk about glyphosate, then in our body, always remember that the production of any amino acid, the synthesis, the biosynthesis of anything, it can be the replication of DNA also, right from nucleotide level up till protein level. Always remember, there is not just one pathway, there is not just one mechanism, there is not just one method which leads to the production of anything, whether it is the replication of DNA, whether it is the production of protein. There is a balance, and there is a and there is always interaction. So if our body is made in such a way that if some pathway is not working, or some mechanism is not working, or some procedure is not working, there are 10 other ways of correcting it, or there are 10 other ways, alternative ways of producing it. So there is always, you can, so that is why when you're doing research, if I found a gene X, and this, I cannot say that this gene X is involved in causing cancer. I cannot say that this gene is, or suppose I have invented a drug, discovered a drug X, I cannot say that this drug can cure cancer, because there are so many other things which are also involved in cancer. There are so many other mechanisms which are involved in cancer. So just saying vaguely that this drug can cause cancer or this drug can prevent cancer is, would be very lame to say, because there are many other things. So saying that this herbicide can have other effects, so the only effect of, let's say, glyphosate is that it will reduce the biosynthetic pathway. So even if suppose uh, this herbicide has accumulated to some extent in the pathway, in the plant, and you have taken up that plant that has herbicide, your body will not have any problem, because the plant is healthy, and that herbicide which is present in the plant, if you say that that herbicide can cause some disease in animals, it is not possible, because that herbicide is made against the plant physiology, and when you are consuming, it is going inside an animal physiology, and plant physiology and animal physiology are different. And even by, say, by some 0.001 chances, there is some similarity in the physiology, then there are so many alternate ways of the production of same aromatic amino acids, that there would be no much problem in it. Okay, I think we should quickly try to complete the questions now. Okay, the pair of hormones required for callus differentiation are ethylene, auxin, cytokinin, auxin, abscisic acid, cytokinin, and gibberellin. What are the
B.
pair of
Oxygen
hormones?
and cytokine. B.
Yes, auxin and cytokinin. Then in plant tissue culture, the callus is regenerated into complete plantlet by altering the concentrations of callus is generated into, see, same question, same, just it is the method of writing is different. Callus is generated into complete plantlet means callus is getting differentiated. Callus is getting differentiated with the help of hormones. That
Hormone.
means the answer would be hormones.
Hormones.
Then next question is, small chemical groups on the antigen molecules that can react with antibody is, on antigen what is present? We have just discussed. On antigens it is
Epitope.
Epitope.
epitope. Yes. Then next question is, what is the function of aminopectin in hybridoma technology? Which pathway it blocks? Specifically, think of a specific answer because there can be two answers which are correct. It blocks which pathway? Aminopectin?
No.
Option A and
A.
C.
Hmm. So option A and C can be correct, but if we think about more specific, so it blocks nucleotide synthesis, but because
No. A.
specifically it blocks the de novo pathway. Then last question, yeah. Animals are selected for breeding on the basis of all of the following except mammary system, ratio of yield to diet, having long ears, disease resistant. So which of the criteria you think is not important in selection of during breeding?
Option C.
Yes, option
C.
C. Have, it doesn't matter. Disease resistant, it is helpful for the proper growth and healthy, for having a healthy offspring. Ratio of yield to diet is also very important because if in less diet it is giving you high yield, it is always good. Even if more diet it is getting less yield, it is of no use. Mammary system and other is important because it is helpful for the nurturing of the offspring and proper physiological function of the offspring. So the point which is not important at all is having long ears. So from with this we complete today's class. So just remind me, in the next class we'll have a very quick discussion about the hybridoma technology, the question that has been raised. But again, I would say that the nucleus status would not really have any effect on the efficiency or anything, because ultimately what we are taking is the antibody. But when we are learning any concept, when we are reading any new thing, we must have all the idea, and we must know the exact mechanism and exact thing which are happening. So we'll try to look onto that aspect as well. And I think then next week would be your last class. We'll, I think, then wind up, and then I think then you have exams now. Then, so okay, we'll meet in the next class then.
No.
Bye.
Nothing. Okay, ma'am. Thank you. No.